Module 8, Objective 2, describe the sensory organs of taste, trace the gustatory pathways to their destinations in the brain, and explain the physiological basis of gustatory discrimination. Gustation, our sense of taste, is going to provide the brain with information about the different substances that we place into our mouths. Gustatory epithelial cells, or taste receptors, are located within taste buds, and they're distributed on the superior surface of the tongue and portions of the pharynx and larynx. They're associated with epithelial projections, called lingual papillae, that exist on the surface of the tongue. We have four main types of lingual papillae. Here they are. The filiform papillae. These provide friction to help move substances around in the mouth. They don't contain taste buds, but they're what makes our tongue rough. So these are located throughout, and they do not contribute to the sense of taste. Fungiform papillae contain at least 5 taste buds each. The vallate papillae may contain upwards of 100 taste buds, and the foliate papillae additionally have taste buds. Taste buds contained an epithelial cell or stem cell called the basal epithelial cells. In addition, they have the gustatory epithelial cells. These gustatory epithelial cells extend taste hairs. These are small microvilli through a taste pore, and they survive about 10 days before they need replaced. They're innervated by cranial nerves that synapse in the solitary nucleus of the medulla oblongata, and the information travels to the thalamus, and then from there to the gustatory cortex of the insula. Remember, the insula is that little island of cortex located between the temporal and the frontal/parietal lobes of the cerebrum. Here we have some of these papillae. The filiform, remember, no taste buds. They're there for friction, so they make our tongue rough to help the tongue manipulate food and move it around. And then we have the vallate, the foliate, and the fungiform papillae, and these all do contain taste buds. And if we look really closely, the taste buds are located down within these grooves, and as food or liquids move down into these grooves, they're going to bind the taste hairs, these little microvilli that are located within the taste pore. And as they bind, they're going to ultimately generate a graded potential, which, if strong enough, will convert to an action potential. And then that's going to, of course, be sent to the central nervous system. We have 4 primary taste sensations: sweet, salty, sour, and bitter. In addition to that, we have umami. This is a savory taste that is imparted by glutamate, which is an amino acid often associated with meats. So umami is a very meaty type taste. And then we have a taste sensation for water, and this is located within the pharynx among the water receptors. In terms of those different tastes, salt and sour chemicals, they simply diffuse through leak channels. So here we have sodium associated with the salty flavor of foods, and then we have hydrogen ions, which are associated with sour or acidic types of foods. And they simply leak through channels, and as they leak, they depolarize the membrane, and again, if that graded depolarization is strong enough, you'll generate an action potential. Sweet, bitter, and umami receptors, these are a little more complex. The food substance or liquid binds to an inactive G protein, causing an activation of that G protein, and then that's going to lead to the activation of a second messenger. We introduced second messengers in the first module of M8O2 when we talk about the endocrine system. But it's worth noting here, since we did bring it up in the last objective as well as this. A second messenger occurs when the first messenger, in this case it would be this food item that's triggering a receptor, when it can't get through the membrane. So basically it's sort of like a relay. The first messenger binds and it causes the activation of a second messenger, which will then carry that signal, that message across. So sweet, bitter, and umami receptors activate via the second messenger system. So the activation of the G protein here is ultimately going to lead to the activation of a second messenger, which will carry the original message to its destination. In terms of taste sensitivity, it differs among individuals. There are many conditions that are inherited that allow for this variation, these differences. But the number of taste receptors began to decline at around age 50. And again, one loses sense of taste along with the sense of smell later in life. Gustatory discrimination, the dissolved chemicals that ultimately bind with the taste hairs, are going to bind to receptor proteins in the gustatory cell. Here we see a stimulus, and in this case we're looking at a food or some type of liquid that is going to bind to the gustatory epithelial cell. And again, if that graded depolarization is strong enough, it will lead to an action potential which will be sent straight to the central nervous system for interpretation. The end result of the stimulation of a taste receptor is the release of neurotransmitters by the receptor cell. Dendrites of sensory afferents wrapped by the receptor membrane is going to pick up those neurotransmitters, and you get the generation of action potentials via the afferent division straight to the central nervous system for interpretation.